Hey folks, we are back. We are ready for our third nervous system lecture video. Last time we met, we had just finished up talking about graded potentials. And now we are going to talk about action potentials. Action potentials are the electrical signals that are going to travel in the axon. So an action potential is an electrical signal that would start in the axon hillock and travel all the way down the axon. Just like graded potentials were waves of change in the membrane potential in the axon, just like graded potentials were waves of change in the membrane potential in the dendrites and soma, an action potential is a wave of change in the membrane potential in the axon. So graded potentials start in the dendrites, travel down the dendrites and soma, and get to the axon. Action potentials start in the axon hillock. Now, they're only going to start if the axon hillock is depolarized to that magic threshold potential that we talked about last time, which had a value of what? Negative 55 millivolts. Now, graded potentials, one of the characteristics of them was the fact that they got smaller with distance. This is not the case with an action potential. If an action potential starts at the axon hillock, if it starts right here, it is not going to stop. It is going to go all the way to the end. There are no big action potentials or small action potentials. There are only action potentials. They are said to be all or none. They are all or nothing. So if you get the axon hillock to depolarize to the magic number, the threshold potential of negative 55 millivolts, you'll get an action potential. If you don't get it there, you won't. An analogy that's often made is knocking over a row of dominoes. You hit that first domino, knocks over the second, the third, and the fourth, all the way to the end. Action potentials are like that. All right, let's explain and try to explain both of them a little more. So we've seen this picture before. We have totally seen this picture before. It is showing us a synapse up top here. We got a synapse right here. We have an axon of a presynaptic neuron synapsing with a dendrite of a postsynaptic neuron. We went through this already. We know that an electrical signal is going to come down this axon and get to the axon terminal. We know that it's going to prompt the release of neurotransmitters from inside the axon terminal to outside into the synaptic cleft. Once they're in the synaptic cleft, those neurotransmitters are going to bind to the receptors here. And the receptors are neurotransmitter gated ion channels. And the binding of the neurotransmitter is going to open these channels, causing sodium ions to then rush through. When sodium ions rush through, the interior becomes more positive, and we have a depolarization. We saw this graph. Start rest at negative 70. When the neurotransmitters bind, we depolarize, and our line gets more positive here. We called that an excitatory postsynaptic potential. Now, in this discussion right here with this picture, we are depolarizing just this spot where the neurotransmitters are binding. We're not depolarizing anywhere else. But earlier we said that depolarization is going to travel. That's going to cause the next spot to depolarize, which is going to cause the next spot to depolarize, and so on, and so on. And that is what we're talking about here. That depolarization travels. So when the electrical signal arrives at our axon terminal here, it prompts the release of neurotransmitters right here. So neurotransmitters are going to be released from right here. Neurotransmitters bind to our receptors, and then sodium is going to rush in. and that depolarizes this spot right here. But that depolarization does not stay put, it spreads. And the arrows here depict the spread of the depolarization. That spreading of the depolarization is called a graded potential. Notice the arrows get skinnier and skinnier, lighter and lighter as the depolarization spreads. And we established that already. We said the depolarization wave of a graded potential gets smaller and smaller as we travel. Now, why does that occur? When the sodium rushes in, there was already a lot of potassium in here. There was a bunch of potassium here. Now, do sodium and potassium, quote unquote, like each other? No, they don't. Does anybody know why? They don't like each other because they have the same charge. Remember, opposites attract, right? Like charges repel. So when sodium rushes in right here, the potassium that was originally here is going to move away. Now, when that potassium moves away, it causes this next spot right here to depolarize. But when new potassium arrives at spot number 1, it causes potassium that was hanging out there to go to spot number 2, which then makes the potassium hanging out at spot number 2 to go to spot number 3, and so on and so on. And this movement of potassium is actually the spreading of depolarization. Okay, but why is it getting smaller and smaller? Well, when the potassium moves, when sodium shoves potassium onward, and then potassium shoves another potassium, which then shoves another, not all the potassium stay inside the cell. Some of them leak out. And because some are leaking out, that is why the depolarization gets smaller and smaller and smaller. But it does travel. And if the initial change right here is big enough, if enough sodium rushes in right here originally, then the depolarization wave, even though it gets smaller, it can make it to the axon hillock and get that axon hillock to reach that threshold potential, which is going to generate an electrical signal in the axon, and the electrical signal in the axon is called an action potential. Okay, awesome. So we know, I should probably erase those drawings there. There we go. We know that we start a graded potential somewhere, like right here. That graded potential then travels, but it does get smaller with distance. Does get smaller with distance. And we hope that by the time it gets to the axon hillock, it is of sufficient size to depolarize the axon hillock, to the threshold potential. And what was the value of that again? Negative 55 millivolts. And if we get there at this spot here, then the axon will generate an action potential. So that is what we're hoping happens. Okay, let's keep going. Reiterating here. We got it. If we generate the signal here, if we generate the signal here, we got to get that signal to travel and travel and travel and get the axon hillock to have enough of a depolarization to get all the way to the threshold potential. It won't cut it if it gets short. So it would take a big old depolarization right at this spot here in order for it to travel all the way, and not diminish so much that we can't get to the threshold here. Does that seem kind of hard? It is. But there's a workaround. There is a workaround. There's something else that can happen. And that's what we've got to explore. All right, check out this neuron over here. Check out this neuron. Here's the soma right here. And each of these points is a dendrite. Bum, bum, bum. Each of these points is a dendrite. And the axon hillock is labeled axon hillock, and the axon is labeled axon. Okay, you with me right there? Cool. Now, I have three axon terminals synapsing with my neuron here. I got synapse A, synapse B, and synapse C. All three of these synapses have the capability of affecting this neuron right here. If synapse A, if neuron A here releases its neurotransmitters, we generate an EPSP. If neuron B releases its neurotransmitters, we generate an EPSP. Remember, an EPSP was a depolarization, right? If neuron C releases its neurotransmitters, we generate an IPSP, an inhibitory postsynaptic potential. Remember, a hyperpolarization. Now, what we're doing is we're measuring what's happening at the axon hillock. This is our voltmeter here. We're measuring what's happening at the axon hillock when neurotransmitters are released here or here or here. Now, I want you to look at the graph. Look at the graph with me. We are starting our membrane potential at -70. Neuron A, that's why they have the A here, neuron A is releasing neurotransmitters. So they're released, they bind to this receptor, the channel opens, sodium rushes in. The sodium rushing in causes a depolarization right at this spot. Then the depolarization wave travels and travels and travels and gets to the axon hillock. And then we measure it at the axon hillock. It's traveling really, really fast, by the way. We measure at the axon hillock and we see the depolarization wave goes up and it wasn't very much. Dies back down. It did not get us to that magic threshold of -55. Okay. So A, doing its release, didn't do enough. And then we do the same thing again. Synapse A releases neurotransmitters again, and sodium channel opens, sodium rushes in, depolarization wave travels. And lo and behold, we get the same thing. Little depolarization, but not enough to get me to threshold. But now we are going to do something different. We are going to have the back-to-back release of neurotransmitters here. So an action potential, a signal is going to arrive here. It's going to cause neurotransmitters to be released, but then another one arrives just a fraction of a second later, and even more neurotransmitters get released. With even more neurotransmitters, we get even more sodium. Even more sodium comes in, which means our gradient potential is even bigger. And look, by the time we get to the axon hillock, now the gradient potential, the depolarization was able to be big enough in order to bring us a threshold. And we see this rapid upstroke and downstroke of the membrane potential at the axon hillock. When we see this shape right here where it goes up like this, back down, down a little too far, then back, that means an action potential happened. That means that our axon hillock generated an action potential, and it went all the way to the end. So what happened here? The two events at synapse A were added together. We added together the two depolarization waves. We added them together. And when we add up subsequent depolarization waves, one right after another, from the same synapse, this guy right here, from the same synapse, we can generate a big enough gradient potential to get us the threshold. And that sort of combination of gradient potentials is called temporal summation. So we can add them together to get our axon to threshold, to generate its own signal. Okay. Fantastic. But you probably guessed this was coming. I had more than just neuron A. We know if neuron A releases neurotransmitter, we get a depolarization, but it's not enough. If neuron B releases neurotransmitter, we get a depolarization in our axon hillock, but it's not enough. If neuron C releases neurotransmitter, we get a hyperpolarization, taking this the opposite way. And what can we do here? We could have neuron A and neuron B both release their neurotransmitters and generate a graded potential right here and a graded potential right here. And then those graded potentials are going to add together and be enough to get our axon hillock all the way to threshold. Now, this type of adding together of graded potentials, when there are multiple neurons involved, is known as spatial summation, because there's two different spaces at which the graded potentials are being developed. Now, notice over here, we could also have neurons A and C both release their neurotransmitters at the same time, and they would basically cancel each other out, because one's depolarization, the other's repolarization. All right. So neurons are doing this all the time. It is rare you are going to get like a single stimulus to a neuron. It is going to get lots of stimuli. It combines the graded potentials, and we see whether that combination is enough once it's traveled to get the axon hillock to the magic number, which is the threshold potential. All right. What do we still need to figure out? We got to figure out what exactly is happening in an action potential. Okay. I have my neuron up here. This is its nucleus, of course, in the soma. Each of these little lines here, these little points here, is a dendrite. Okay? And this is my axon hillock right here. I'm just going to make an H for axon hillock. Boom, boom, boom. And this whole tube here is my axon. Now, what we are going to assume is that we are going to generate a graded potential or two. Let's say we generate enough graded potentials that they're going to add together and get my axon hillock to that magic threshold value. Okay? They're going to get my axon hillock to that magic threshold value. Okay? And the first spot we're measuring is right here in the axon, okay? Where that electrode is going in. And what we can see at that spot in the axon is we have a depolarization, then a subsequent repolarization, and then a subsequent hyperpolarization. So there's some D, there's some re, and there's some hyper, okay? That's what happened at electrode number 1. So if we get our axon to the threshold potential, we are going to cause the axon to have a depolarization, a repolarization, and then a hyperpolarization. All right, but that's not it. Once those three things, once the D, the re, and the H happen at spot number 1, what's going to happen at spot number 2? Well, check it out. At a moment later in time, spot number 2 experiences a depolarization and then a repolarization, and then it will experience a hyperpolarization. And then a moment later in time, look at spot number 3, I mean. Number 3 is going to do the same exact things. Then spot number 4 is going to do the same exact things, and so on, all the way down the axon. So, an action potential, it is a wave of 3 things. It is a wave of depolarization, repolarization, and hyperpolarization that happens down the entirety of the axon, from the axon hillock all the way down to the axon terminal. And that wave of these 3 events prompts the axon terminal to release neurotransmitters. So, we're starting to establish what an action potential is. And remember, our whole goal here, our whole extraordinary goal here, is to figure out how a neuron sends a signal from one end all the way to the other. From your lower back, where your longest neuron starts, to your bottom of your foot, where it ends. And we know we are going to have graded potentials in our dendrites, in our cell body, and then in our axon, we're going to have action potentials. Now, the action potential is a wave of change of these three events that travels down the axon. Okay, let's not stop. Let's keep on going. We can have a slightly more nuanced graph. If I go back for just a quick sec here. Whoops, can I go back here? I wanted to go back. There we go. If I go back for a second here, on my graph here I said we had a depolarization, a repolarization, and a hyperpolarization, and that repeated itself. We're going to zoom in on one of these little trios, okay? So let's do that. Here we have the three changes in an action potential. Notice we're starting at rest here, starting at rest. This is where the graded potential arrives. The graded potential is big enough to get us the threshold. At threshold, there are special channels called voltage-gated sodium channels. They cause sodium to rush into the neuron, into the axon. And that is what's causing this upstroke, this depolarization phase of the action potential. By the time we get to the peak, sodium channels have closed, and voltage-gated potassium channels are now open, and so potassium rushes out. Positive charge leaves, makes the inside more negative, bringing us back down. Bring us back down. And we even go so far back down, we have a period of time where we're below normal resting. So we have the repolarization phase, and then we have the hyperpolarization phase, and then we are back to normal. Okay, this is what's happening in my axon. Reaching threshold causes voltage-gated sodium channels to open. Sodium rushes in, causing the depolarization. That same magic number threshold potential also caused the momentary subsequent opening of voltage-gated potassium channels, which caused the repolarization as potassium rushed out. And we had a little extra hyperpolarization here, which we'll talk about in a moment. Okay, good stuff. Complicated, but good. So I got a little spot on my axon hillock here. And I am starting with that axon hillock totally, utterly at rest. Now the axon has all the proteins we've mentioned so far. All the proteins we mentioned, in the last video. So it's got sodium potassium pumps. It's got sodium leakage channels, potassium leakage channels. But the axon has its own special channels. Its own special channels are these voltage-gated guys. Voltage-gated sodium channels and voltage-gated potassium channels. At the resting membrane potential, these guys are both shut. So no sodium is going through these guys. No potassium is going out these guys. These guys are both shut. If the graded potential arrives and is big enough to get the threshold, it is going to cause both of these guys to open up. So, starting at rest, we're now going to look at what happens when the graded potential has arrived and these guys have been affected. The sodium channel opens up immediately. The potassium channel is slow. It's creaky. It responds slowly. So the sodium channel is open right now, but potassium is, the gears are working, but it's still closed. Because the sodium channel is open, we have the ingress of sodium, the influx of sodium, which is depolarizing this spot right here. And this is why our graph has this upstroke, this depolarization phase. Sodium is open, sodium channel is open, potassium channel is not yet open, but the gears are working. And the changes in both these guys are due to the arrival of the gradient potential that was big enough to get the electrical state to change to the threshold potential. Okay, so now we got to talk about what's happening after we get to this peak here. By the time we get to this peak, enough time has passed for the sodium channel to now have closed and the potassium channel to finally have opened. And when the potassium channel has opened, potassiums here are going to rush out. The egress of potassium, the efflux of potassium is positive charge leaving, making the interior more negative, and we have our downstroke here. Now, I want you to notice something. I want you to see something here, okay? If we go back for a second, when they were both originally closed, notice it's not the ball and chain that's closing the sodium channel. It's these hot dogs here. The hot dogs are closing the sodium channel, not the ball and chain. The ball and chain is just hanging. But the hot dogs are keeping these guys closed. Now the sodium channel is open. The hot dogs have moved. The ball and chain is still just hanging out. And now that I said the sodium channel was closed and the potassium channel is open by now, it's not the hot dogs that closed it. It's the ball and chain. And when the ball and chain closes the sodium channel, that sodium channel is inactive. That sodium channel is turned off. Now, it'll only be turned off for a fraction of a second, but this matters. The sodium channel is inactive for a moment because of the ball and chain that closed it. Okay? So, potassium channels are open, sodium channels are turned off at this spot right here. Remember, we're only talking about this one spot right here. All right, we don't know what's happening at the other spots yet. Okay. All right. Remember how the potassium channels were slow to open? I said they were creaky. The gears were going. Well, they're also slow to close. And so there's this extra period of time where they're starting to close, but some potassium is still leaking out. And because some potassium is still leaking out, that is why we have this little hyperpolarization phase here where we get a little more negative than the original rest. Okay? Now, during this hyperpolarization phase, the potassium channels are in the process of closing, but they're still a little bit open. And check out my sodium channel. The ball and chain is no longer there, and it's the hot dogs closing it, which means my sodium channel is now closed, but it is not inactive. And this will have ramifications later on, okay? So don't forget this. All right. If we were in class, I would assign you, one of you, as like the person who can't forget this. And then I would call on that person later on and expect them to tell me. It's too bad we can't be in a real class right now. It's much better. All right. Awesome. If we look at that graph again, we started at rest. Rest. Gradient potential arrives right here. Gradient potential gets us to threshold. At threshold, boom, immediately. Sodium channels, voltage-gated sodium channels open up. Sodium rushes in and we have our upstroke, our depolarization phase. By the time we get right here, sodium channels have closed, but it's the ball and chain, okay? It's the ball and chain which makes them inactive. Meanwhile, the slow, creaky potassium channels are open. Potassium rushes out. Potassium is going to, that potassium efflux is going to cause this repolarization here, all the way back down. And because potassium channels close slowly, we get this little extra period because a little extra potassium left, and then it's the hyperpolarization phase. Now, if we think about it for a second, we think about our sodium channels, our voltage-gated sodium channels. When we're at rest, our voltage-gated sodium channels are closed. And they're closed until we get to threshold, okay? Then at threshold, the voltage-gated sodium channels are going to then open up. And they're going to be open up for this. They're going to be open for this entire period of time. But by the time we get to this spot right here, until we get to this spot right here, so the entirety of number 4, sodium channels are now closed, but they're not only closed, they're inactive because it was that ball and chain that closed them, okay? So in spot number 4, they're closed and inactive. By the time we get to spot number 5, the hyperpolarization phase here, we're back to just being closed. And then when we get back to rest, we're still just closed. So closed, open, closed and inactive, closed, closed. Okay, don't forget about this closed inactive here and the fact that we're just regular good old closed at all the other spots. So check out the directionality here. We get spot number 1 to experience the depolarization, the repolarization, and the hyperpolarization. Then spot number 2 experiences the depolarization, the repolarization, and the hyperpolarization. And then spot number 3, spot number 4, and so on and so on. The depolarization at spot number 1 is what's going to get spot number 2 to threshold and cause the trio of events to happen. The depolarization at spot number 2 is what's going to get our spot number 3 to threshold and cause the trio. The depolarization from spot number 3 is what's going to get spot number 4 to threshold and cause the trio, and so on and so on. This is how it just keeps on going. It just keeps on going. It is not just channels opening in one spot like it was for a graded potential. It is this domino effect where depolarization here causes channels to open, and we get more depolarization, and then that depolarization opens channels here, and that depolarization opens channels here. Whoops. And so on and so forth. Okay. If we zoom in on our axon, this is our axon's plasma membrane right here. I see these purple guys here are my voltage-gated sodium channels, and these two are open, which means sodium is rushing in, which means this spot right here has depolarized to positive 30. Now, what's going to happen is the fact that the depolarization is going to travel down my axon, and it's going to depolarize the next sodium channel, causing this next dude to open up. Sodium rushes in right here. The depolarization then travels onward to the next one, opening it, and so on and so forth. And this is how the action potential travels. The depolarization at one spot, so this one spot right here, will then cause the next spot to depolarize the threshold, which will then cause the next spot to depolarize the threshold, and so on and so forth. But there is a potential problem here that we have to address. Maybe you thought of it. It's okay if you didn't, because I'm going to tell you. When we are depolarizing this spot right here, this spot gets to positive 30, right? Because the sodium channels are open. I just told you the depolarization travels this way down the axon and depolarizes the next sodium channel, getting him to open up. But does the depolarization know it's supposed to go just down the axon that way? No. The depolarization will also travel backwards. Will it open this sodium channel? If it did, that would be terrible. Because if this depolarization caused depolarization here, then depolarization here would cause depolarization here, and we would get this vicious circle. But it doesn't go backwards. Because remember there was that momentary stage when the sodium channels were not only closed, they were also shut down. That ball and chain turned them off for a moment. And because of that inactive state, for a moment, the action potential cannot reverse. Action potentials are one way. They don't go two directions. The textbooks often say action potentials are unidirectional. Unidirectional, which is a fancy way of saying one way. And they're one way only because of the fact that the sodium channels inactivate for a moment. Okay. Awesome. We can see the potassium channels doing their things as well. Potassium channel here is closed. This guy's open, so we're going to repolarize here, which means in a moment this guy's going to open up, and we're going to repolarize the next spot, and so on and so on. So there is the wave of change of D, re, and hyperpolarization. That's what an action potential is. All right. I got this graph here again. Beautiful graph. One of your homework assignments is actually to draw this graph for me so you know all the parts. So you could do that right now. You could pause the video and sketch this graph. Hey, good idea. Okay. I'm jumping ahead a little bit. You know what? I forgot to do something. I thought I forgot. I want to do it right here. Let's talk for a second here. Remember this period of time right here? This period of time right here. What was going on with the sodium channels here? The sodium channels, they're closed and what? Inactive. So during that period of time, the sodium channels are closed and inactive. Bear with me for a second here. You remember that, right? You got that? I want to jump back for a second here. So bear with me. This guy right here. This guy right here. Closed. Inactive, right? Yeah. We said the good thing was the the action potential couldn't like turn around and go backwards and get a vicious cycle going. But there's another thing I want you to think about here. Suppose this guy opened. This guy could open. He's in a resting state. Suppose this guy opened, sodium rushed in, and the depolarization traveled to this guy. Could this spot then open if there was a depolarization wave traveling this way? No, because it's in that inactive state. So there is a limit on how many action potentials can travel along a particular axon. There's only so many that can travel along a particular axon. And it's because of this inactivation state, the ball and chain. And there is actually a word, a term, for the period of time, during the repolarization phase, there is a term for this period of time right here. This period of time right here, number 4, that like lightish blue-ish, that is called the absolute refractory period. R-E-F-R-A-C-T-O-R-Y. The absolute refractory period. And that is the period of time where we absolutely, like no way, no how, we absolutely cannot have another action potential. Whether we're trying to go forward or trying to go backwards. Okay? In normal like English, the word refractory means stubborn, so like resisting action or resisting change. So it kind of makes sense here. That refractory period is where we are resisting change. We cannot undergo another action potential. Okay, all right. There's another refractory period though. Yeah. The other refractory period is this period of time coinciding with the hyperpolarization phase. So number 5 here. That number 5 is known as the relative refractory period. So remember, absolute refractory period is a period of time during which absolutely no new action potentials can happen. During the relative refractory period, it is just harder for an action potential to take place. There's relatively more resistance to another action potential happening during this period of time. Why is it harder for another action potential to take place during this period of time? Well, the answer is because during that period of time, check it out, we are farther from threshold. So we're hyperpolarized. We're farther from threshold. All right. Amazing. We are doing fantastic today, and we're almost done. I know this is another long-ish video. Look at this neuron for a second with me here. It's a beautiful neuron. Got some gorgeous dendrites, soma, axon, right? Absolutely. What we've been talking about for a long time is how an electrical signal travels from here. Trying to explain how it gets all the way to here, right? That's what we've been doing. We talked about how if we bind some neurotransmitters here, we get a gradient potential, which could be a traveling wave of depolarization, which if it gets to my axon hillock, there we go. Maybe I can turn that into an action potential, and the action potential will travel all the way down. We talked about how the action potential was a wave, right? We said it was a wave of a trio of events, a wave of depolarization, repolarization, and hyperpolarization, DRH. Wave DRH. And we looked at a graph of those events, right? We looked at a graph, didn't we? There it was. This is the graph of the 3 events that happen at every spot on our axon. So every spot on our axon is going to have an identical D, Re, and hyperpolarization. So every single spot, is going to have a D, Re, and hyperpolarization, and they look exactly identical. So depolarization right here will look just like right here and right here and right here and right here and so on. Okay? No matter where we did that graph, it would always look the same. Now, this begs a question. The question is, how are we actually sending information? How are we actually sending information if every action potential that starts here goes here, and every action potential looks exactly the same? All right, I want your brain to chew on that for a minute, okay? I want your brain to chew on that for just a second. okay? All right, I got my neuron here. I'm going to talk about this neuron and this picture down here for a second while you keep that question I asked for you in the back of your brain. Okay, let your subconscious work on it. So here's my neuron. There is my axon hillock right there. Beautiful axon hillock right there. And there's my axon. So let's say we generate an action potential. So we're going to get a wave of depolarization, repolarization, hyperpolarization all along this neuron, right? And we're zooming at the axon, and we see our voltage-gated sodium channel. Stimulus here is sodium rushing in, which depolarizes this spot. The depolarization then meanders onward and causes the next channel to open. And then sodium rushes in right here. And then this depolarization meanders onward, opens the next channel. Now, notice that this neuron's axon is unmyelinated. It does not have any myelin. What that means is every little spot, it says unmyelinated, every little spot on the axon is going to undergo the trio of events, D, re, and hyperpolarization. Now, what we got to do is contrast that type of action potential traveling, which by the way is known as continuous conduction, with a myelinated situation. So, if we jump to a myelinated neuron, similar neuron, multipolar neuron, again, it's got dendrites, soma, there's the axon hillock right there, axon terminals, of course, at the end. But this guy's myelinated. It's got a myelin sheath. Look at all these Schwann cells, right? And we're zooming in right here, and we're zooming in on the axon. We can see these two Schwann cells. We can also see the gaps between Schwann cells. Each gap is called a node of Ranvier. So there's a node over here, there's a node right here, and there's a node over here. And what's going to happen, what you're going to notice is we only have the channels at the nodes. So sodium is going to rush in here, depolarize this spot. The depolarization then travels all the way to the next node. Depolarizes these guys. They open, sodium rushes in, the depolarization travels, and so on. Just like in continuous conduction, but it's only happening at the nodes. Now, here's a question. How come when the depolarization happened right here, how come it was able to travel all the way to the next node without getting so small that it failed to get to threshold? The reason we're able to travel this distance here is because of the insulation. So one of the things the insulation does is it lets our action potential, the trio of events, happen only at the nodes. A millimeter is actually kind of far. If it weren't for the Schwann cells here, if this were just a naked axon and the channels were this far apart, the graded potential would die out. The depolarization, rather, not the graded potential, the depolarization would die out. Now, what's the upside of this? The upside of this, by only doing the depolarization, repolarization, hyperpolarization events at the nodes, we don't have to do as many of them. And this type of action potential travel is a lot faster than the continuous conduction that we just talked about a moment ago, a slide ago. And by the way, in myelinated neurons like this type of action potential movement is known as saltatory conduction. Saltatory means jumping, because early physiologists were like, the action potential is jumping from node to node. It's not actually jumping, but that's the idea. It's the analogy. So we want this situation in neurons whose speed is of the essence. So neurons that control movement, neurons that relay information about your balance, about your proprioception, neurons that relay information about pain. We want myelinated neurons. We can't have every single neuron be myelinated though, because there would be a space cost. I mean, those Schwann cells take up room. And there's also an energy cost. Those Schwann cells are living. They need oxygen, glucose, etc. But we do myelinate neurons that matter. And myelination allows us to do the action potential's trio of events only at the nodes. And that is called saltatory conduction. All right. Guys, we're almost done. I swear. Look, we got this slide, and now we got two more. That's it. That is it. Okay. I got a synapse in that box right there. And there is an action potential happening in this axon up here. The trio of events is happening at the nodes. And then happening along the telodendron, all the way to the axon terminal. So here's the telodendron. So the trio of events is happening all the way down this telodendron, all the way down here, getting to the axon terminal. And here's where things get a little weird. When the axon terminal depolarizes the threshold, when we get this guy to depolarize the threshold, we don't open voltage-gated sodium and potassium channels anymore. The axon terminal, we find voltage-gated calcium channels. And when they open, calcium rushes in, which is going to depolarize the axon terminal. But even more importantly, the influx of calcium causes these little bags of neurotransmitters, synaptic vesicles, to undergo exocytosis. So if an action potential arrived here, calcium channels would open, calcium would rush in, a couple of these guys would undergo exocytosis, releasing neurotransmitters onto my neighboring neuron, causing it to be stimulated. Okay, this is really good. We have now traveled from one end of a neuron to the other. We have now talked about how binding of neurotransmitters right here generates a gradient potential. That gradient potential travels to the axon hillock, diminishing along the way, but maybe adding to other ones. And then if this axon hillock gets the threshold, we generate an action potential, and the action potential travels all the way to the axon terminal, prompting the release of neurotransmitters onto another neuron, causing the same thing to happen in it. So hey, now we know how a neuron gets a signal from one end to the other. Now you know that. I understand you might need to review and study a little bit, but now you know. Okay? You're on the way. You're on the pathway to knowing. But hey, let's figure out one more thing. Do you remember I asked you that question a moment ago? If every action potential looks alike, if it's an all or nothing phenomenon, how do we actually communicate information? Well, it's not about the size of an action potential because there's no variation there. Instead, it's about the frequency. The more action potentials you have, the stronger you stimulate the next neuron. So if I had back-to-back action potentials going down this axon, right at one after another, I would release more and more amounts of neurotransmitter, causing an even greater number of gradient potentials on the next neuron, and they could sum together. So, stronger stimulus, if you poke your skin, harder you poke it, the more action potentials you're going to send. And the greater the intensity of that signal. Because you sent more action potentials. You didn't send bigger ones, you sent more. All right. This is so good. We've only got one more to go. One more slide. There's one more thing we got to do. We talked about how the axon terminal releases neurotransmitters into the synaptic cleft here, the space here. And we know the neurotransmitters bind to the postsynaptic cell, causing channels to open and a new graded potential to happen. But what we haven't done yet is figure out what happens to those neurotransmitters after they're released. We also haven't named any neurotransmitters yet. There are a ton. I got good news for you. I think we're only going to talk about like 2 specific ones later on. If you ever take like a real neuroscience course, a real neurophysiology course, which I totally recommend, you will learn about a whole bunch of different neurotransmitters. But for now, to end today, we got to figure out what happens to the neurotransmitters once they're released into the synaptic cleft. Well, there are 3 things. My favorite is depicted right here. The cell that released them, the axon terminal that released them, just has a transport protein that brings them back in, and they get repackaged and recycled. They could also be taken up by neighboring glial cells. They could also diffuse away into the blood and just travel somewhere else in the body, maybe broken down by the liver. They could also be broken down by enzymes in the synaptic cleft, perhaps on the postsynaptic cell. Now, why do we got to do this? We can't have the neurotransmitter just hanging out forever. In fact, when neurotransmitter grabs onto the channel protein, it doesn't stay on it permanently. The channel protein will let go of it. And it might grab onto it again, but it might not, because one of these 4 things could have occurred. So we cannot constantly have the neurotransmitter in here. Now, a lot of drugs do work by affecting these processes. There are drugs that mimic the actions of neurotransmitters by binding to the same receptor and causing them to open. There are drugs that grab onto the receptor but don't do anything, preventing the regular neurotransmitter from doing its thing. There are drugs that block this reuptake, so the neurotransmitters persist in here for longer. There are drugs that block the enzymes, so that the neurotransmitters don't get broken down and persist for longer. All right, guys, with that, amazing. We are so done with our Intro to Nervous System section, which is great. I am happy we're done with it. It took us 3 kind of long videos to do it, but that's okay. Hopefully you watch them. Hopefully you got to the end of this one. Hopefully you watched it. All right, I feel like I should give my extra credit stuff at the very end of the videos. Never in the beginning, always at the end. That is a great idea, isn't it? If you're hearing this now, you'll know. All right, bye-bye.